Yes, hi everyone. I am Nazli Ayhan. I am a researcher in Aix Marseille University and I'm working in National Reference Center for Arboviruses in France. Today, I would like to talk about you how to discover new viruses in Sandflies. When most people think about the beginning of AIDS, they are going to think back to the 1980s. And certainly this was the decade in which we discovered AIDS and the virus caused the disease HIV. But in fact, this virus crossed over from chimpanzees to human many decades before in Brazil, Congo in 1920s. So what took us so long to discover the virus and identify the viral agent? Of course, there are many reasons behind these questions, and this is not unique to HIV. Today, we discovered many viruses, and 60% of emerging infectious diseases are of animal origin. Recently, many viruses have been discovered that can be transmitted from animals to humans. But we are focusing on the top of the iceberg after they have already been adapted to human. And they are difficult to address, as we have seen in the case of HIV. Can we change and identify them earlier and understand all these movements of the viral agents? Of course, it's possible, but we should focus on other steps. Within this virus, there are also some arthropod-borne viruses, which means that the viruses can be transmitted by arthropod vectors. It can be mosquitoes, ticks, or sandflies. Arthropod-borne viruses transmitted by sandflies can be grouped into 3 different viral families, and they are distributed worldwide, as you can see in the map. But in today's talk, I'm going to more focus on Phlebovirus family and the genus Phlebovirus. They circulate in the old world. Many properties of sandfly-borne Phleboviruses make them good candidates for emergence. Different Phleboviruses can co-circulate into the same biotope, and they are transmitted by the same sandfly population. Their distribution is geographically driven by the components vector, and therefore the climate changes have a direct impact on the ecology of Phleboviruses. So, as you can see here, they can co-circulate, and the climate change has an important effect on the circulating Phleboviruses. There are many factors can affect the emerging of viruses. They can be human activities, animal activities, or environmental factors. And as we've seen in the SARS-CoV-2 epidemics, for example, the easiest way to transportation, it caused a emerging of SARS-CoV-2 epidemics recently. When we look at the sandflies, there are small insects. Both males and females feed on sugar sources in the wild, but only females take blood meal prior to laying their eggs. Unlike mosquitoes, they attack silently, and they are mostly nocturnal, but some species can bite during the daylight. Sandflies are distributed throughout the world in tropical and subtropical, arid and semi-arid areas and temperate zones. And they show a peak during the warm season. So the question is how to discover new viruses in sandflies. The very first step, collect sandflies, a lot of sandflies if possible. The higher the number of the collection, the probability to identify and discover new viruses is getting higher. So sandflies can be collected by using light traps, sticky traps, or aspirators. For virus studies, we prefer to preserve the sandflies alive until we put them in the cold chain. That's why we prefer to use the light traps and aspirators to keep the viral integrity. And then sandflies are preserved in the cold storage, if possible in the field work in dry ice, and in the lab in -80. Or if our aim is not to isolate the virus, we can also keep the samples in the RNA stabilizing solutions to maintain the viral integrity. After bringing them, the sandflies into the lab, collected samples are sorted by date, region, species, sex, and feeding statute. Why it's important to separate these? Because if we can identify the sandfly species morphologically or molecularly, we can identify the vector species who can transmit the virus. So that's why it's important for us to identify the sandfly species. Also, we are separating them by sex, because if we identify the virus in male individuals, which may indicate the virus can transmit it transovarily, which means that from infected mother to the eggs, or ventrally during the copulation with the infected female or male. We are also separating the blood-fed female individuals, because afterwards we can do the blood meal analysis to identify the host species. What are the following steps? In the lab, afterwards we can do the virus nucleic acid testing. We can do metagenomic analysis, or we can do virus isolation that I am going to give more details about. So virus nucleic acid testing includes RT-PCR assays. But if we don't know what is the virus, we haven't discovered yet, how we can use the RT-PCR systems to identify the virus? So the response is we can use pan-PCR systems. A pan-virus PCR assay is a molecular diagnostic test designed to detect a broad range of viruses in a PCR reaction. So while designing the pan-PCR systems, we are targeting the conserved sequence region in one genus or in one small group in the genus, which can amplify multiple virus species at the same time. Here there are some examples. We have some Phlebovirus PCR assays which can target viruses like dengue, Zika, yellow fever, or West Nile to use in arbovirus surveillance. And we have other pan systems such as pan-coronavirus assays that can target different coronaviruses. For Phleboviruses, there are several pan-Phlebovirus assays are available in the literature, which are targeting different segments of the Phleboviruses. It can be S segment or L segment. Recently, we performed a comprehensive evaluation of these tests to see the sensitivity of the tests with Dr. Ceylan Polat. And we saw that Matsuno assay is one of the most sensitive pan-Phlebo assay for Phleboviruses. Because the pan systems are generally less sensitive than the virus-specific assays. Recently, with technological developments, we can also use metagenomic assays in which we are sequencing all the genome, which can help us to discover completely novel viruses by analyzing all the genetical material present in the samples. But since we are sequencing everything in the sample, we can have some host contamination. Sandfly or bacterial sequencing can obscure viral discovery. Short reads lengths make it hard to assemble the complete viral genome. Or we can have some false positives. This can overcome with manual verification and confirmation of the novel viruses. One of the best tool is to isolate the virus, because if we isolate the virus, we can also do further analysis with the virus. How we can isolate the virus? We have different cell lines coming from different origins. We have Sandfly cell lines, human cell lines, pre-domestic wildlife animal cell lines are present. So after homogenizing the Sandflies, we can take the supernatant and inoculate them on the different cell lines. After a incubation period, we observe the cells under microscope to see if there is any cytopathic effect or not, which means that if the cells are dead or not. Because when the virus is attached to cell surface, they get inside the cells and grow in the cells, and they explode them and try to find other cells to infect. But not all viruses are cytopathic. There are some viruses can grow in the cells without killing them. So for this reason, after inoculating the samples on the cell lines, rather if they have cytopathic effect or not, we are testing all the supernatant samples by using RT-PCR assays to be able to identify if there are any virus has been isolated or not. We found a new virus. How we know that this virus is a novel virus species? According to latest International Committee on Taxonomy of Viruses, ICTV, criteria for Phlebovirus genus, amino acid distance for RNA-dependent RNA polymerase gene should be lower than 5%. After doing the genetic analysis, if our sequence has a lower distance than 5%, which means that it belongs to one species. If it's higher than 5%, it means that it's probably a new virus. And when we look at the phylogenetic relationship between the serocomplexes for old world Sandfly-borne Phleboviruses, we can group them into 3 serocomplexes: Sandfly fever Naples serocomplex, Salaba serocomplex, and Sicilian serocomplex. As it's mentioned in previous webinar by Professor Remy Charel, there are several pathogens in these groups. In Naples groups, we have 2 human pathogens. One of them is Naples group, which causes 3 days fever. And we have Toscana virus, which can cause central nervous system infections. And in Sicilian serocomplex, we have Sicilian virus, which can also cause 3 days fever. In Salabat complex, there is one virus has been identified in only one case as a human pathogen for now. So the current knowledge is still limited for the pathogeny of this group. The global structure of the Phlebovirus phylogeny was like this before 2009. And this is the current status of the phylogenetic tree. During last decades, many different Phleboviruses have been discovered, especially in Mediterranean basin in this example. And it's getting increased with the recent studies. So we have done field studies and we discovered a novel virus. What are the next steps? If we manage to isolate the virus, we can do seroprevalence studies to address if the virus has a medical importance or not, whether in human or in animals. We can perform some in vitro and animal model studies to characterize the virus and identify the pathogenicity in different animal models. We can organize clinical studies and develop diagnostic tools. And afterwards, we can transfer these diagnostic tools to keep on the surveillance for this virus. So I'm going to talk about some real studies to have examples. Recently, one Phlebovirus has been identified in Sandflies in Tunisia, which was closely related with Toscana virus, which raises the question, is Punic virus capable to infect humans or not? And how to distinguish different Phleboviruses from each other using antibody studies. Because when the viruses are closer, the genetic relationship between the viruses, the greater the cross-reactivity of antibodies. So this was a challenge. And the question was rather, what are the respective proportion of seroprevalence against Toscana virus and Punic virus in humans in Tunisia? There are several serological assays that we could have used in this study. There are ELISA tests are available. ELISA tests are quite sensitive, but they are less specific. So that's why in the first step, we performed ELISA test, and afterwards we tested the samples with seroneutralization assay, which is more specific. In this study, 1,273 human sera were tested from different regions of Tunisia. With ELISA test, we found that 516 sera ELISA positive. Afterwards, we include these samples with also including some negative samples by ELISA and perform seroneutralization assay for Toscana and Punic virus. As a result, we saw that 516 Toscana positive and 91 Punic positive. 216 sera without any antibodies, 7 sera equally affinity for both viruses, 21 sera higher affinity for Punic virus, and 509 sera higher affinity for Toscana virus. What are these results telling us? So, which means that the 4% have antibodies against Punic virus and 96% have antibodies against Toscana virus. So, as a conclusion, from the Phleboviruses, 96% of infections are due to Toscana virus, which means Toscana virus is the main pathogen with impact on public health in Tunisia. And Punic virus, with only 4% of infections, should not have an important public health impact. I would like to give another example from my thesis work. We have collected Sandflies in different countries in Balkans, and we identified 2 different Phleboviruses. One is from Croatia, which is called Phlebovirus, and the other one is from North Macedonia, which is called Bregalaka virus. They both within the Salabat group. And after isolating these viruses, we performed a virus seroneutralization assay from the samples collected from North Macedonia for Bregalaka virus. And we saw the seroprevalence is the 3.3% in human and 2.4% in dogs. Afterwards, we performed a virus genetic study to be able to see the virus, if rather the virus is able to grow and the genetic of the viral growth. We used 2 different cell lines. One is a green monkey African monkey kidney cells, and the other one was mosquito cell line C636. As you can see in the graphs, we saw that the virus is capable of growing in mammalian cell lines as we expected, but also grow in mosquito cell lines in C636 cell lines, which was an interesting finding because normally sandfly-borne Phleboviruses are not capable of growing in mosquito cell lines. Afterwards, we look more closely to phylogenetic tree, and as you can see here, there are two viruses within the same cell complex are from Culex mosquitoes, which may raise a question whether this group has a mosquito-borne Phlebovirus origin or not, or whether the mosquitoes can transmit these two viruses or not. So these are questions waiting for answer. Afterwards, we performed an artificial infection with using 2 to 3 day-old suckling mice. We infected them intracerebrally with Bregalocavirus and Zabavirus. As you can see in the table, we couldn't observe any signs or symptoms for the mice infected by Bregalocavirus, but some mice show some symptoms for Zabavirus. We also euthanized them between day 6 and 10 after infection, and we tested the brain samples to see if the virus is present in the brain cells or not. As a result, we saw that we managed to detect the viral RNA of Zabavirus and Bregalocavirus. Here another example from the Sandflies in Albania. We couldn't manage to isolate, but we detect a virus which is called Balkan virus clustered together with other Naples virus group Phleboviruses. After detecting these viruses, we designed a specific real-time primers to be able to screen all the samples collected from different Balkan countries. And as a result, we saw that Balkan virus not only circulating in Albania, but also in Bosnia-Herzegovina and Croatia. So you can see in the map, the red points represent the virus positive sandflies. In recent decades, there are many articles has been published in Mediterranean basin from Turkey, Greece, Iran, different countries, and they reported the novel Phlebovirus presence in different countries. But not only in Mediterranean basin, I would like to give another example from Kenya. Sandfly-associated Phleboviruses has been detected from sandflies. It is called Entepes virus. They also performed a virus genetic and characterization experiments to see whether the virus is susceptible to grow in different cell lines, and they saw that the virus has a broad susceptibility in different cell lines with a peak genome copy number in swine and rodent cells. As you can see in the first graph, the virus cannot grow in C636 cell lines, which is a mosquito cell lines, but in sandfly cell lines, it can. They also performed the artificial infection with using 3 to 4 day-old Swiss albino suckling mice, and they observed symptoms. Also, the mice were dead between 5 and 8 day post-infection. They also perform a seroprevalence assay And they saw that 13.9% of samples has neutralizing antibodies against Entepes virus. Women and men were infected at equal rates, and the detection rate didn't differ between healthy and febrile patients, because this group was including some febrile patients and blood donors, but they couldn't detect the viral genome in the serum samples, which suggesting no causative link in the present samples with Entepes virus. But the detection of neutralizing antibodies suggests the widespread of previous human exposure and infection of Entepes virus in Kenya. The virus discovery is not only limited with Phleboviruses, but also other Phleboviruses has been detected and isolated from Sandflies. I put an example here. A novel clade in genus Phlebovirus has been identified in Sandflies collected from Ecuador. And in the photos, you can see that it causes a cytopathic effect in C636 cell lines. So why virus discoveries are important? In 1971, Toscana virus was isolated in central Italy from two different species of sandflies, Phlebotomus perniciosus and Phlebotomus perfiliae. But it took us 15 years to identify this virus as a pathogen. So virus discoveries are important to understand the virus origins. It's important to develop vaccines and treatments, developing preventing measures, and monitoring evolution and the ecology of the virus in nature. And as we know here, what happens in Vegas doesn't stay in Vegas. So this virus circulation is not local circulation. So it's impacting everybody, as we have seen in SARS-CoV-2 epidemics. Thank you for listening.